In this video, we will discuss about a paper that has been recently published, which shows that the CAR-T cells can be engineered in vivo using this targeted lipid nanoparticles that they have designed. So we will discuss about that paper in this video. We will discuss the first figure where it shows its specificity that it can target the designated site and does not expressed in the liver. So this is how the structure of this lipid nanoparticle looks like. It has this targeting binder that decides at which locations these lipid nanoparticles need to be expressed, and it has this ionizable lipid nanoparticles and the mRNA payload. So first, they designed this lipid nanoparticles, optimized the lipid nanoparticles that they call as L829 targeted lipid nanoparticles, and then they checked its expressions compared to the lipid nanoparticles that is used in the mRNA vaccine. So in the case of when they use the control of PBS, they do not see any signals. When they use the lipid nanoparticle that is used in the mRNA vaccines, they can see the signals in both the places, that is liver and spleen. But when they use these targeted lipid nanoparticles, not targeted, they are optimized lipid 829 nanoparticles, it is not expressed in anywhere, as it is not been targeted to any of the sites. And then they use these targeted lipid nanoparticles, commercial lipid nanoparticles with targeted towards the spleen, and they can see the signals even in the liver and also in the spleen. And when they use this lipid 829 targeted lipid nanoparticles, they can see the signals on specific signals in the spleen with the minimum signals or almost negligible signals in the liver. So this indicates its specificity. Then they wanted to check this clearance time in the liver, and compared to the benchmark, they can see that their lipid 829 nanoparticles cleared within 72 hours, and even after 7 days the expressions remains low, whereas compared to the benchmark lipid nanoparticles, its expressions remains high in the liver. Then they checked it in the rat and the mice which are sensitive to the lipid nanoparticles. So for the rat, when they check it for the acute phase proteins expressions, when they are treated with the lipid nanoparticles, this commercial total targeted lipid nanoparticles that is targeted to the CD5, and this L829 targeted lipid nanoparticles which is targeted to the CD5, and they can see that expression of these acute phase proteins are high in the case of their benchmark targeted lipid nanoparticles compared to their optimized CD5 829 targeted lipid nanoparticles. And similar results they have seen for in the monkey also, where they can see that the signals for the benchmark acute phase proteins are high compared to the case where they use their optimized CD5 L829 targeted lipid nanoparticles. Then they, and in that case they see the signals, or they have injected the mRNA of this luciferase protein, so they can clearly see the signals. And in the panel B also they use the luciferase proteins instead of this mRNA for this mRNA payload. Then they targeted this CD8 antibodies using L829 targeted lipid nanoparticles using this mCherry. In this case, panel F, they use this mCherry as the reporter proteins, and they can see that when they use the CD8 L829, because from the literature they are able to find out that targeting CD8 plus T cells is more beneficial for some diseases compared to the CD4 T cells. So they decided to optimize the system for CD8 T cells. So when they use the CD8 L829 targeted lipid nanoparticles mCherry signals, they can see that it is expressed only in the CD8 plus T cells, but not in other T cells, neither in CD4 plus, neither in monocytes or in B cells. Or we can say that the signals are very minimum in this case. And For when they check it in the spleen also for the different types of T cells, they can see that signals are high. Only signals are high for the CD8 T cells compared to the CD4 plus T cells, indicates that the specificity of their CD8 plus L829 targeted lipid nanoparticles is good, and it can be used to develop these CAR-T cells in vivo within the uh, in vivo, that is within the patients, instead of developing these CAR-T cells ex vivo and then transfuse it into the cells. So this is how they validate the specificity of their optimized targeted lipid nanoparticles. We will discuss in subsequent videos about the further results, how they prove their CAR-T cells engineering in the in vivo is working, and what are the safety concerns that they have addressed in this paper. So that's all for this video. If you need to understand more about their experiments or this topic in detail, you can go through the reference that has been provided in the description. Thank you for your time.